All right, so this is our second of the great not so British Bake Off. This is uh, a concept that is based off of cooking shows that you might see like online or today, where the idea is you go into like a kitchen or somewhere and you actually cook through something with someone. So like kind of start to finish, you're actually in your kitchen. That's right. And so for today, in our first kind of season, if you will, is looking at DDA, label free quant, and this started off as the focus was grad students, but I think it's applicable kind of more in general. And it's not just that we're going to learn how to use some tools, but we're also going to get to meet the people making the tools. And so for today, I'm not really joined, but across the country is Phil Wilmarth. And yeah, Phil, if you want to say some introductions.
Okay. I've got kind of an unusual background for proteomics, so I'll just kind of give a real quick little bio. I grew up on a dairy farm in Tillamook County on the Oregon coast. I went to a very small high school, and then went to a small liberal arts college, and then went to graduate school in the chemistry department at UC Berkeley, studying nuclear chemistry. I worked for Dr. Glenn Seaborg, and entered graduate school in '81, got my degree in '88, and then postdoc for 4 more years at Lawrence Berkeley National Lab. My wife and I were both looking for small college teaching positions. I didn't get any job offers. She got a position at Reed College here in Portland, Oregon. We moved here in '92. And then after a while, not finding any other work, I started working in bicycle shops, and then eventually worked my way up to the parts and accessory purchasing manager at River City Bicycles, which is one of the biggest single store bike shops in the country. And eventually got a little burned out on that, decided I wanted to get back into research, and started volunteering in a lab at OHSU, Oregon Health and Science Universities, doing proteomics, and eventually worked my way up to research assistant professor, and now I develop the data analysis tools for our proteomics core.
So we go back to that. So because I think the timeline is really fun with proteomics. So you kind of came back to OHSU. What was that, late 90s?
2003.
Okay, 2003. So things were, we had, was that about the same time that Sequest had come out, right? Or
yes, well, Sequest paper was 95.
Okay.
So that was pretty established, and Thermo had BioWorks. We had a LCQ Classic instrument in Dr. Larry David's lab, studying aging changes in human lens proteins.
Mm-hmm.
And then at some point we got LTQ off the instrument grant, and then that produced so much more data than the LCQs. Things were really struggling to analyze the data, and that's really when I started working on my processing pipeline. But,
that's amazing.
Yeah, I had no background in proteomics or biology, so I've never had a biology class. I've never taken any biochemistry, computer science, statistics, informatics, none of that. So all self-taught.
Well, and that's kind of what I think is fun about that time is that the field wasn't really, I mean, there weren't people that were necessarily trained in that. Like now you can kind of come into it and it's all pretty fresh. But I mean, I think I was getting things confused. I mean, FDR, I think the Elias paper, right? Like that was, even statistics on shotgun proteomics was just kind of at the time being figured out, right? I mean,
well, that's 2007 for the Gaijin Elias paper.
Yeah.
Things. Mud pit stuff was 99 or so. Mm-hmm. We were doing some early mud pit experiments with the LCQ, producing giant data sets, 50,000 total MS2 spectra.
And so
quite a difference from today.
And so the inception of kind of your pipeline, did it just kind of really start slow, and then, or was there kind of a moment where you're like, okay, this is 2008? It kind of
started 2006. So we got the LTQ in sort of fall of 2005.
Mm-hmm.
And then the data sets were just so much bigger than we were getting off the LCQ. We'd been using a little bit of Bioworks and then also Scaffold, early Scaffold. And both really had struggles with the LTQ data sets on the Windows XP computers, where you had a lot of resource limitations, not a lot of RAM, not a very fast multi-core CPU situation, and then spinning hard drives quite a bit slower than the current solid states. So all of that was quite problematic, and just to load all of the big data sets in. So the pipeline approach I developed is, if you could identify, set your FDR thresholds and get rid of all of the matches that aren't any good, your data set's about 10 times smaller, and then you're kind of back to where you were before the LTQ. So then you could make data analysis work on the computers with the resources we have. So the fundamental thing there is to try to figure out where to set the score thresholds and just get rid of all of that data that doesn't pass the score thresholds, and just focus on the stuff that's relevant to the stuff. And like Scaffold brings in and keeps all the IDs with the different probabilities, so you can dynamically change the thresholds and stuff. But you don't get that option with my pipeline. You decide the cutoffs ahead of time, and then your protein report's going to be based on that. And if you want to go back and change it, you have to start over.
Yeah. Well,
but it saves resources.
Yeah. So today we're gonna go through the pipeline, right?
Yeah.
So I guess in pause, I asked you this before. I mean, this is your initials, right?
Right.
Okay.
Yes. It's an analogy to in the very early days from CERN. When I started graduate school, there wasn't an internet. It hadn't been invented yet. And very early on, there were some abilities to share files with BitNet and other types of technologies. And the big national labs sort of had ways that their computers could connect. But general, small computers having abilities to connect remotely were pretty limited. But CERN had these tools, so they had a physics analysis workbench, PAW, and they had a set of tools for that, so you could be able to visualize and analyze high energy physics data from CERN. So that was kind of where the PAW thing came from, as an acronym for a data analysis toolkit. And then it just happened to be my initials also. So it was kind of like, became a proteomics analysis workbench thing to be able to use my initials.
Nice. No, that's see, things I would not know.
Yeah. Many years ago, I basically spent my graduate student days sitting in front of computers that you'd now see in a computer museum. So, good times.
Yeah.
You want me to start screen sharing and get going?
Yeah. Yeah. Let's see.
Okay.
See how we start.
Okay, so I'm going to minimize this window to make the cool effect go away.
It is fun.
All right. So basically, my software and kind of everything I have done over the years is all sitting at GitHub now. So to find that, just github.com/pwilmarth. Type. Okay, and sometimes people wonder about where does that short name come from. And back in the old days, 8-character usernames and email addresses were pretty common. So it was pretty frequently you'd use like the first initial of your first name and then 7 characters of your last name. So that's where the P. Wilmarth comes from.
That I think I asked that question once.
Yeah, because my Twitter handle, P. Wilmarth, was already taken, so I had that H. So it's P. Wilmarth. Twitter. Okay, so this is my main GitHub page here, and we have start here repository, which has a kind of a roadmap to the whole site. What we basically have are some blogs I've written about some proteomics topics. We've got some collection of software I've written over the years for doing proteomics analysis. We're going to be talking about the PAW pipeline today. There's also utilities for FASTA file stuff and some downstream processing tools as well. And then I've also got a bunch of data analysis of public data I've pulled down using Jupyter notebooks and the sort of showcase quantitative proteomics and statistical comparisons in a visualization-rich way that's also reproducible and makes nice supplemental files that you can put into data archives. So lots of examples there, and they're all sort of described and detailed. And then there's some meeting content, talks, and things I've given that I've kind of turned into content here on the site. So that's all available.
And I have to interrupt you because I have to say what I think a lot of people would like to tell you, which is it's an amazing resource, right? I mean, it's kind of having this compendium, and you've got it like the navigation, start here, and then you can kind of walk through it. And even I was looking up something today, and it's linking out to the Jupyter notebooks, and it's really impressive. And I think so. Yeah, thank you. I think a lot of other people would say thank you too.
Yeah, GitHub's much more than just a place to stick your source code, but there's a lot of things you can do with it. It's a pretty powerful environment. And like they sort of say, if you write software and you don't have good documentation for it and good ways for people to understand how to use it, what's the point of having written a software in the first place? So I kind of took that to heart and tried to thoroughly document stuff. But it's grown over time, and it's a lot to maintain. I'm pretty much a group of one, so it's getting pretty overwhelming to try to keep things up to date. But let's go to the POP pipeline. So we've got all of the software here. Everything's publicly available, open source, freely available. If you want to download the code, you can either clone the repository if you know your way around Git, or you can just download a zip of the content of the repository and then open the file folder up. The nice thing about the Python scripts here, there's no installation process per se. As long as you have a Python installation on your computer, you can run the Python scripts in a variety of different ways. So they can live in a folder anywhere on your computer. No formal installation process for that. We do have some software dependencies, some tools that the pipeline calls. We'll look at that. So the documentation kind of describes those things, gives an outline of the pipeline, and has some different navigation things here, but we can go to the dependencies part. And the things that we need to be able to do, we have to be able to access our binary instrument files, the raw files. So MS Convert. MS Convert is an open source package with a variety of vendor software libraries that can be called to access the instrument files and convert them into formats that can be read by downstream tools. We'll be using the open source Comet search engine. That's what the pipeline's built around. And then you also have to have a Python 3 installation. It's easier to just use the Anaconda scientific Python distribution because that includes NumPy, Matplotlib, and Pandas, which are third-party scientific Python tools that are not part of the standard Python distribution. So the easiest way to get all of those things installed and working is just download Anaconda. And there's links here for different things, so you can talk about the MS Convert installation and setup. And got a link there to get to the download page on SourceForge. So all of that stuff is available there to help you get the software installed correctly so that the pipeline will run. Okay, so I think at this point I'll start playing with some actual data.
And just because I have this like proverbial person in my mind who like is convinced that things don't work. I mean, these are pretty standard, MS Convert, it's going to stick it in a folder within ProteoWizard, Comet, like these are just things that we're then going to point back at, right? So it's nothing fancy going on here.
Right.
Okay.
Comet comes down sort of like my pipeline as just a zip folder that you open up, and it's got an executable file in it. Mm-hmm. So there's nothing that it installs in a particular place. The ProteoWizard is a little more formal installation and actually installs in a folder in your system files. So you kind of have to sort of know where those things are and then basically point the scripts back to them. Mm-hmm. And I've got a little description about how to set up sort of some Windows batch files in a folder that you put on your Windows search path so that you can more easily create sort of a one level of indirection to where those tools are installed. You don't have to worry about putting in a specific path to the actual tool itself. You put in a path to the batch file. So it's a way to kind of decouple. You can update your Proteo Wizard package more easily that way without having to mess with the script.
Oh, so you won't have to like change it to folder 3.02 or something.
Right.
Okay.
Right. So the script just points to the bat file, the Windows batch file, and the Windows batch file points to the actual executable for MS Convert. So then when you want to change to a different version of MS Convert, you just change the Windows batch file pointer. That way you kind of decouple. Okay, so I made a little folder here with California sea lion data, and at some point here I'll show that we've got a full data analysis repository just for this data set. It's available on GitHub. So if you're interested in this, you can then check out that repository and also see the spectral count quantitative comparison.
I mean, I'm a big fan of the California sea lion.
It's a fun data set. Let's see. So I've got a folder here where I just have one raw file because the conversion steps and the Comet search steps take quite a while on this data set. So we're not going to have time to look at all of those, but I'm just going to pick one file here to show you how these scripts run for that. And then we'll finish up with some analysis that's got kind of all of the 19 files. Okay, so that's what's in this special folder. And normally on project organizations, you make a folder called raw files and you put the raw instrument files in that. And that's kind of the starting point for where the scripts are going to go. And it'll produce additional files and new folders, but all in the same folder where your project sort of lives. Now, Anaconda is installed here. We can look at that. And there's this Anaconda prompt, which is a nice thing that you have to do. Anaconda comes in and it installs who knows where on the computer, but if you go into the Anaconda prompt here, it's got those paths defined so that it can find various Python things. And if you just open a Windows command file and tried to do the command I'm going to do next, it wouldn't run. It would say file or command not found. But we get some additional system path stuff defined here when we're inside this Anaconda prompt. Idle is a programming environment that's part of the standard Python distribution. So it's got a console window and it allows you to open up source code windows and then run scripts and test and debug them and all of that. This kind of environment where I developed the tool, and it's I like for running the tools because the console window stays up and you can look at it, scroll back on it. You can even copy and paste out of the console window if you want to get kind of the detailed information on things. And if you run stuff through kind of the Windows command line, when a script finishes, then the command window closes and you don't see all of the console output and stuff. So the persistence of the console window and idle is one of the things that I like.
And can I say something just for someone that's listening around? So
Sure.
kind of what you just said is, Anaconda is a super easy, it just installs a bunch of stuff, and then you're going to have this other Anaconda console. What you're saying is, if you type in Python or idle from Windows, unless you've set that path, it's just going to be like, what are you talking about? Anaconda has those paths set, which means if you type in idle to Anaconda, it knows because it's like, oh, you obviously mean this. Right.
So here it's just a standard Windows command idle. It's not recognized.
And
so somehow you've got some different search path things added when it's the Anaconda idle, Anaconda prompt.
Yeah.
And things like if you want to do Conda updates and things like that, the Conda package manager, that also generally needs to be through those idle command windows.
I'm super lazy. I just use the Anaconda prompt for everything.
Yeah, you could run these scripts too from a variety of different ways. So you could do it from Spider within Anaconda or PyCharms, another environment people like. I just kind of go with this standard idle one because it's part of the standard Python distribution. So kind of no matter what sort of environment you're on, it's usually available. But Anaconda, because they do these other IDE environments, they kind of hide idle from you. They'll make it very easy to execute it. So you kind of have to do that Anaconda prompt and then type idle to get to it. But basically here you've got the console window, and then we want to open our scripts. Here I downloaded the pipeline files from GitHub as a zip archive, decompressed it, and then stuck the folder in here with the other Bakeoff files. So this is a copy of the scripts that are at GitHub. And the first one we want to do is our MS Convert GUI. So we've got basically a little GUI application that does the file conversion. That's the first step. Then we've got a little GUI program that sets up and runs Comet. And then we have a GUI program that looks at the score histograms and sets up the FDR analysis. And those things all have GUI in their name. And then we make our final reports with the PAW results program. And those are kind of the basic ones that we're going to run today.
Mm-hmm.
So MS Convert GUI. And the Python source code shows up, and it's documented, and you can actually learn something and play around with it. But generally speaking, we just open these scripts up and run them. So we can do that a couple of different ways, either run module from this menu or just do the F5 key module. Okay, now we get a little GUI window, and we can browse to our raw files. Select the file. The pipeline also has support for TMT labeling, which is what most of the radio buttons and things are here for. We're basically just going to make our MS2 scan data so we can identify the peptides, and we can usually live with centroided data with thermal instruments without any problem. So most of the defaults here are just fine for this. So once we got the raw file loaded, we can just hit start conversion. And it's going to start going. And this says we've launched an MS Convert call. And we also see that we've now got an MS Convert process running. We can open that Windows command up and look at the progress. This will take a couple of minutes to run. While we're doing that, let's get back over to GitHub. So up here, we've got kind of a little diagram of what the pipeline steps are. So here we're reading the raw files with MS Convert, and we're producing MS2 files, which they're sort of like the MGF files if you use Mascot. It's a file format developed by the Yates Lab back in 2003. It's just sort of a text version of the peak lists. And if we had TMP data, we would also get reporter ions. Yes, I still have a landline in the Good. And then after we get the MS2 files, then we'll be running Comet, and it'll produce the search output files and also a top hit summary file. So that's kind of where we're at in the pipeline right now. I don't know if you got any questions.
Yeah, I mean, I think something. As users, I think we know about a lot of tools, but we rarely, I think, have gone and used them. I know for me, like we've been in the same room, we haven't actually met in person, but like when we first started interacting, it sounded very complicated, all these steps. But most of your tools are made, you've got GUI wrappers around pretty much all steps. And if it's not all steps, there's pop-up windows, right? I mean, I'm not, to use these things, I'm not hard coding. I'm not writing these big, command line things out. And I think for me, I felt really stupid for not trying it sooner. I was like, wait, these and they're not flashy, but they work, right? Like, even this schematic that you have, like every one of those steps, it works. It makes the thing. It outputs it. And so just for me, something, if you're watching along, even if this looks complicated, when you actually try it, I don't think it is complicated, right? I mean, there's not a lot going on here, right?
Right, and it's a real pipeline. So they're separate steps. Some of these things could be kind of wrapped together if you always did the same kind of analysis, but there's some variations on data analysis you can do by having it be modular like this.
Mm-hmm.
So it's a little bit more flexible this way, but for some of the real basic applications, it seems like it's kind of, some extra windows and things you have to click on that maybe one button could have done it all. But like you say, there's kind of nothing magic there. And computer technologies and programming environments have changed a lot over the years. So I'm pretty old school. And when I started, it was things like having a Perl installed on your computer. You did some Perl scripts for things. And then Python came along, and it was still in that sort of environment where basically you download and install a language on your computer and then write some scripts to do something. Now things get a little bit more fancy. You put things in containers, or you got these one-click big black box type applications for things. But this is still kind of the old school where you just string together some scripts and do the processing. And I've tried to hide a lot of the command line stuff. I'm a terrible typist, and I prefer clicking on things with the mouse. So we kind of have these GUI wrappers around things. So now
I'm a big fan.
Our conversion is finished. So we make this text file from MS Convert. It looks a lot like an MZML file without the XML tags, and we write it as a gzipped file to save some space, and it's quite a bit smaller than the original raw file because it's just got the MS2 scans in it. If we go up one level, we see now that we have an MSN files folder that's been created, and there's our MS2 file. And these files are pretty large, but if you have a programmer's editor, you can open them up, and they're all text files. They're readable. You can uncompress the text GZ file here if you want to browse what's really in these sorts of files to know how your parsing of that file needs to be written. So everything here is really working off of human readable files, although they can be large at times. But you can really kind of dig in and understand every step if you want to take the time. Okay, so that is done. So now we're ready to go to the comment step. Again, we just run the script, F5 or the run menu. Now we get a dialog, kind of a form thing here for the comment parameters. First thing we can do is select a FASTA database file. And I'll put that in here as well, the California Sea Lion RefSeq. That's there. The pipeline, we do wide tolerant searches, and I'll talk about that when we're looking at the delta mass histograms. Here, the data was actually done with high resolution MS2s, but we're going to leave this demo at low resolution because that runs faster. The urine samples, also semi-triptych, is a better choice for identifications, but that also takes a lot longer to run. So we're going to leave that as fully triptych here just for demonstration purposes. We've got oxidized methionine as a variable modification in there by default. Generally speaking, we want to have as few PTMs specified as possible. The pipeline is really designed for very simple types of searches. We do have static modifications we can look at. So here we've got the standard reduction and alkylation with acetamide. Mass shift there. We can also get static masses for TMT tags. Click those buttons. So the 57 on cysteine was good for this data. So now we've got everything pretty much specified. And then we need to save the Comet params file before we can launch our Comet searches. So we have to go find that MSN folder, select it, and then they'll write the Comet file in there. Now if we look at MSN, now we got a Comet params file in there in addition to the MS2 file. Then we can run Comet. And again, the console window tells us we started Comet, and now we see we've got a Comet Windows command window up. First thing it has to do is load the spectra. These files have about 70,000 MS2 spectra per LC run, so it takes a while to load that in. And we see the search here is going to take about 3 minutes to run at this rate, but it's chugging away. Let's see if we've got anything else back at GitHub that might be useful. Okay, so we've made the conversion step, we're doing the database search. One of the things that's kind of interesting about Comet, Comet is the open source version of Sequest written by Jimmy Ng, and it's available and maintained by University of Washington. And Sequest, when it originally started, when it had PTMs, they were specified with special symbols. So the first PTM you specify would get an asterisk, the second PTM would get a pound sign. There was no information about what the PTM was or what the delta mass associated with that PTM was in the peptide sequence string. The peptide sequence string would just have this special character after the modified residue. So that's what the pipeline's built for. And Comet, at some point, Jimmy changed the way the PTMs were reported so it could support more complicated things like these PEF FASTA files that have a whole bunch of PTM information in them and do more complicated searches. And then he started embedding the delta mass value in square brackets adjacent to the residue that's modified, but that completely breaks the pipeline. So we have to use a little bit older version of Comet. I haven't had a chance to update the pipeline to the different way that PTMs are specified, but the sort of old school Sequest form is kind of what everything's designed for. And then one of the other interesting things about Comet, when you've got a peptide that matches to multiple proteins, Comet will tell you the first protein accession in the FASTA file that contains that peptide, and then it'll tell you how many other proteins also contain that peptide, but it won't tell you which proteins they were. So it'll say, here's an accession, here's a peptide sequence, and then it'll say like +5. That means there's 5 other protein sequences that contain that peptide.
Oh, Phil, I think I lost you on that one. I guess we'll wait for Phil to come back as we're going down this road. And if it's me, I don't really know what to do. Maybe I will. All right, we're going to take a little break and see if I can find Phil and come back. Okay, so Phil, we lost our connection. You had just been talking about the earlier Sequest where it would basically assign the first entry it hit in a FASTA and then just say some nebulous, there were 5 other proteins that shared that peptide.
Right. So what we have to do is take that peptide sequence and then take our FASTA file, digest all our proteins, make a dictionary of the peptides, and then look up that peptide and see what are all the proteins that it matched to. So that's what's been occurring. That takes about 6 minutes. The search took about 3 minutes. The lookup here, there's a lot of time up front to digest the whole FASTA file. And then once that's done, the lookups get progressively faster as there's more and more data that gets searched. It kind of remembers what it's already looked up. And you see the peptides multiple times, typically in experiments. So that kind of speeds things up. So there's a little upfront cost, but then it goes pretty quick once it gets going. And here it's just chugging through the different scans that are there, and we got to get up to 70,000 scans.
Wait, will you say that again? So, I mean, I remember late 2000s, we kind of had this parsimony rule, right? Like where we would let a peptide be at one protein. And then somehow in the last however many years, we do this anti-parsimony where things are protein groups. So what you're saying is you're just taking the peptide IDs from kind of old school Comet, and then you're kind of assigning those with this tool. Is that what you just said, or am I getting that wrong?
The protein inference, you have to take all of the different peptides and all of the proteins they match to, and then you invert the process. Now you make a list of the proteins and you collect up all of the peptides associated with those proteins. Mm-hmm. So that's where you get that redundancy, and then you start reducing the redundancy by proteins that have identical peptide sets you group together, and things that have a subset that's a formal subset of something else you get rid of. So we're not quite to the point where we do that, but we need to know the identity of all of the different proteins that contain these peptides to be able to do the protein inference. So that's what we have to do in this lookup step.
Okay, and is the lookup step within, I mean, is that something you wrote, or is that, okay, see things I didn't know.
Yeah, and if you're familiar with like the transproteome pipeline, somewhere in there's this XInteract step that basically has to do the same sort of thing if you're working with sequence type results, because you need to know the identity of all of those protein matches. The newer Comet, I think, has, it's more explicit in its output, so I don't know if you still need to do this. I think it might explicitly list all of the matches instead of just telling you the number that it matched. So this might be somewhat tied to this a little bit older version of Comet that I need because of the way PTMs are specified. But at any rate, that's what we got, and that's how it works.
No, good to know. Really good to know.
So what we now have over here, the MS2 file is basically the peak list that we started with. When Comet ran, it wrote out this companion file, SQT, sort of a sequenced results format. And then we read that, find those top hits, and then look them up in the FASTA database to make sure that we've got protein accessions for all of the matches. And that gets summarized in a top hit TXT file. And everything now is basically going to be just working with this top hit summary file instead of anything else.
But you didn't, when we went off into the nether regions there for a second, you didn't open up a new GUI. This is still at that step,
No.
Comet GUI. Okay.
Right. So that should still be in here somewhere. It hasn't come back to my prompt. So, wait a second. So it says conversions have completed, quit when ready. So quit that, then we'll get our constant prompt back. So now we're ready to go to the look at the histograms. And that's the histo GUI program. Run that. Now we have to select top hit summary files. And what we will do here is actually look at all of the data instead of just that single data. So what we have here.
So we're going to do a little magic of TV action.
Right. So here we've got results from all 19 raw files. What we have to do is select the top hit summary files that are in our MSN folder. We are, select those. Now the default settings here, you can kind of change some things on that, but I never do. So now we just need to load and plot the histograms. Now the first thing we want to look at here are the mass agreements between the sequences that were assigned to the peptides and the mass of the ion that the instrument measured.
Mm-hmm.
And normally we expect correct sequence assignments and the mass of the peptide to agree very well, basically have a 0 Dalton discrepancy for the delta mass. And we'll make a delta mass histogram using the target decoy method. We have to build databases with decoy sequences when we run the pipeline. And we'll see what those look like here in a second. The first thing it does is it reads everything in, and then when it's high resolution data, we skip the 1+ ions and just look at 2s, 3s, and 4s for the ions. We get some warnings here from the matplotlib stuff.
Should I be worried about red text there, or just kind of go with it?
No, they're warnings. At some point they might be hard errors, but that's something that's not really part of Python per se. That's part of the NumPy scientific Python stuff that's been added on. So at some point I'll probably have to look into those things and address that, but they're just future warnings that this may be a problem down the road.
Cool.
Think of these things where when you're doing vectorized operations, kind of like R or something, there's always the question, what do you do if there's an undefined value in a cell, or a divide by 0 or something like that. So this is kind of like how it handles those odd things in the data stream. So they're kind of related to that. But I need to do quite a bit of research on those packages to see what those messages are and how to figure out how to work around them. Okay, so now we got our delta mass histograms here. We have -1.25 to +2.5 range. We do that so we can capture a 1 Dalton region in addition to this 0 Dalton region. The 0 Dalton region is where we expect the majority of our correct matches to be. Matches to our target database are shown in blue. and matches to the decoy half of the database are shown in red. And we see that we have this nice excess blue at 0 Daltons. But we also see here, and this is one of the nice things about the pipeline, it integrates a lot of quality control information. You see that we have 2 peaks at 0 Dalton instead of expected 1 peak. And the data was collected in 2 chunks in May and June. So what's likely happening here is we've got a little bit of a systematic mass here associated with one of the times and not the other. And then over here at 1 Dalton, we'll have 2 peaks. That's now 4 because of the mass shift. So let's cancel this, run it again, and just look at the June data, which is doesn't have this here.
Should talk to the person that ran that data. You know, honestly,
the POP pipeline can handle the doublets. You just set a wider window around them, but it'll be a little confusing to people. So we'll look at cleaner data, which would be the more typical case here, but it'll be half the data.
Okay. But it would, I mean, but yeah, like you said, you can keep with that. This just kind of helps our discussion right now.
Right.
Okay.
It's one of the nice things about the POP pipeline that 0 Dalton peak doesn't have to sit at 0 Dalton. If there's a systematic error, it can sit somewhere near 0 Dalton, and it's still fine. We're just going to set a window around capturing the whole peak, and same thing for 1 Dalton. So there can be some tolerance of that, and then you can even have some mixed data where the calibration isn't the same, and it just kind of broadens these peaks out. But we can just set a little bit wider windows on it, and it doesn't really change things all that much. So here, now we see that we've got a very sharp 0 Dalton peak. We got this wide full range view, and then we've also got an expanded scale around 0 Dalton, expanded around the 1 Dalton region. The 1 Dalton region, we've got 2 peaks here. The first one is caused from deamidation. So that's a 0.984 Dalton mass shift. And the other one is a 1.003 mass shift that's caused by capturing the first isotopic peak instead of the monoisotopic peak on it. And one of the nice things about doing this wider tolerance search is you don't have to specify deamidation, or you don't have to do a narrow tolerance search with isotopic errors allowed. You can just do a search without specifying those extra things and still capture those identifications here. So now what we want to do for each of our charge states is set a narrow window around our 0 Dalton region and a window around both of these peaks at 1 Dalton. And we can adjust these windows, I'll show you how. So here what we'll do is we'll expand the scale. So if we just double click axis there, we can kind of bring that up. And we see that our dotted lines, which are where our thresholds are set by a default algorithm that kind of runs down off the peak and stops when it thinks it's down near the baseline. We could maybe broaden that out a little bit and capture some more of that peak. So what we do is we go down and click in the table to get the GUI focus onto that widget. And then we can specify with the left arrow that we're working on the left threshold, and then we use the up and down arrows to move it around. Now we just broaden it out by going up in the table. And then if we hit the enter, we've now reset that threshold. Then we do the right arrow to get the right threshold, and then we move that down in the table. So we can broaden that out. And now we've captured all of the 0 Dalton peak. Now here, the 1 Dalton peak is pretty good on the left side, but it might need the right side adjusted just a teeny bit. So we can kind of fine tune these things. And we've got a similar situation for 3+ peptides. And typically in these experiments, identifications are about 2 to 1, 2+ peptides over 3+ with trypsin. But we also have a very small number of 4+ identifications as well. The heights of these histograms tells you the relative amounts of these different classes of peptides. And 4+ would recover enough to make it worthwhile, but it might be on the order of 2 or 3% identifications for 4+ with trypsin. So here I'm not going to tweak any of the thresholds. We'll just compute score histograms. So the score histograms are going to be dependent upon these delta mass windows. So we're going to look at our scores just inside that 0 Dalton window, and we'll look at our scores inside that 1 Dalton doublet window. And then we'll also look at our scores for any delta masses that weren't inside either window. So everything else that's in there. Okay, so now we show up with our histograms, and we see that we've got 2+, 3+, 0+, 2+, 3+, 4+ for the 0 Dalton peak, and the same things, 3 charge states for the 1 Dalton peak, and 3 charge states when we're not inside either window. We've got unmodified peptides and modified peptides. Sometimes the modified peptides have a very different search space size, like phosphorylation is a very large search space size compared to the unmodified peptides. And our noise matches the red distribution, our decoy peptides, partition sort of as a function of the search space size. So if we've got a large search space size like phosphorylation, we're going to have a lot of noise in that category, and that's going to require a higher score threshold to get a 1% FDR than our unmodified peptides. Oxidized methionine is pretty close usually in score cutoff to the unmodifieds, but we separately look at histograms for every PTM that you specify. It's one of the reasons you don't want to specify too many PTMs, because it kind of makes the statistics start to get a little bit sketchy. So here we basically have also our fully tryptic peptides and semi-tryptic peptides. This is a search that was done with the semi-tryptic peptides shown, and we see that the fully tryptic peptides in the 0 Dalton region have almost no decoy peptide match. So we can set a very low score threshold and capture nearly all of the correct matches. But the semi-tryptic ones have a bigger search space, they have a lot more decoy peptides associated with it, and we have to set a higher score threshold to get the 1% FDR for that class of peptides. The program here also puts the dotted lines defaults at 1% FDR.
Yeah, a quick question. I mean, first off, something that I've really enjoyed, like talking to people like you, is there's this kind of common knowledge, like there's certain biofluids, or all biofluids, where like semi-tryptic is appropriate, right? I mean, but if you didn't know that semi-tryptic is something that was going on, I mean, is that something that the data can kind of tell you, or is it really something I mean, I always set it based on some experimental knowledge, making that choice.
Yeah, there's no, you'll find semi-tryptic peptides even in a good cell lysate sample rather than a biofluid, because the peptide sequences sometimes are fully tryptic in one protein and semi-tryptic in another protein. So when you're kind of matching that peptide across all the proteins, you'll pick up fully tryptic contexts and also semi-tryptic contexts. So it becomes a little bit tricky. Is the semi-tryptic peptide correct when you did a tryptic search? But in biofluids, we know that there's endogenous proteases, protein degradation, protein processing, removal of signal peptides, all kinds of things that make semi-tryptic peptides. But knowing whether, I don't think there's a signature if you did like a fully tryptic search that those semi-tryptic peptides and you need to go redo your search. I think they pretty much stay hidden. So it's just like you say, kind of a domain knowledge thing.
Yeah. And last one, the discovery score, that's a score from Comet, or are you?
Discriminant score. So it's a linear discriminant function. Pretty much the same functional form that peptide profit had in the 2001 paper for that. So it's kind of a discriminant function where there's a term for the cross-correlation score, sequence primary score, and sort of an equal weighted term that's related to the delta CN term, which is sort of the gap score, the difference between the top hit and the next best hit. And then there's some minor things for like preliminary scoring function and also mass, delta mass that's kind of hiding in there. But those last two terms are very downweighted in the discriminant function. So it's really dominated by kind of equal contributions of this gap score and the primary search score. And this improves the separation between the correct and incorrect distributions. So we see that the incorrect distribution, generally speaking, is pretty well separated from the correct distribution, and that allows us to recover identifications with pretty high sensitivity. And if you looked at score distributions with just sequence cross-correlation score, then these two distributions will be much closer together, and it's much harder to get a 1% FDR and then recover as many of the correct identifications. So this discriminant transformation really improves your error control and sensitivity. Okay, and we've got oxidized methionine. peptides as well. Their score distributions are smaller, but they're pretty much the same shape and locations as we have for the unmodified peptides. And we again have thresholds set by default here. We can go in and tweak these if we really want to, or change the FDR level. Like here, we can see that we're setting it just under 1% FDR is where the cutoff was. Sometimes in samples where you don't have as many identifications, or the experiment isn't such a large scale one, then you might want to set a lower threshold, maybe 2 or 3% FDR or 5% FDR, and you can adjust those thresholds accordingly. It's the same basic thing. You click on the spectrum, you click at a different place where you want the FDR to be, and then if you hit the return key, you'll move the threshold to the green line.
But like in this case,
pardon?
But like in this case, like that's
yeah, this
0.65 FDR or whatever. Yeah.
So 3+ looks similar, and also oxidized methionine. So most of these are going to be pretty good. When we see something like this where we don't have a dotted line set, that means we never hit 1% FDR. So the threshold's basically sitting out here at -6. So we're not really needing to invoke any discriminant score cutoff for that to get our 1% FDR. And if you look at that, it's like we're starting at 0.86%. So, and that just shows the power of using that delta mass approach up front. You've basically eliminated essentially all the noise here in the fully tryptic matches for the 3+ peptide. Spores are the same thing, but we've got fewer of them. Now, if we go to the 1 Dalton region, that's much smaller. The peaks for the 1 Daltons are very much smaller compared to the 0 Dalton. So we've got kind of a constant noise level across that. So the signal to noise is a lot different for the 1+ region than it is for the 0 Dalton region. So our incorrect score distributions here are relatively larger, and that pushes our score thresholds up for these 1 Dalton peptides. But it's the same basic idea. We're just setting the threshold just above the bulk of the red distributions. Oxidized methionines. This is mostly, it's kind of a quality control step. You sort of see what the score distributions look like. If you're looking for, expecting oxidized methionine peptides and didn't see any, you wouldn't see any blue matches in these things. So sometimes people are looking for a particular PTM, and this is a good way to validate it. You can say, most cases, those sorts of special PTMs are usually not present in the samples. And you get score distributions where there just aren't any scores for those PTMs. And you can definitively say to someone, that PTM isn't in your sample. So sometimes that's a really nice feature to be able to say with some confidence, no, it's not there. You need to go do a different experiment. But you get that kind of integrated quality control aspect to the pipeline by looking at these score histograms. Okay, and then that and oxidized methionine. Now here's an example where we had fewer than 50 counts above our 1% cutoff. So any sparse category like this doesn't get a threshold set at all. It's just excluded as a conservative sort of thing. But if we want to play the game of trying to maximize things, then we can just click on the table. It'll show us where the dotted line is just above the decoy distribution. And if we hit the return key, then we get the dotted line on top of the green line. And then we'll be capturing those 27 additional correct IDs. So that's pretty much how you set the thresholds and score distributions. Now, another thing that's rather interesting, a lot of people don't realize there's a lot of peptides that are sequenced and identified that don't have an accurate mass at all. So here we actually have quite a few 2+ peptides that were not inside the 0 Dalton window or inside the 1 Dalton window. And when you look at that wide tolerance delta mass histogram, you don't see any other regions where there seems to be any excess blue matches across there. But collectively over the whole region, they do add up, and they can often be a few percent of the identifications. So this is a way to increase the sensitivity. The instruments often have this thing where, in the old days, I think that if things were quite noisy, it really couldn't assign a monoisotopic mass. or make that call, but it still knew there was an ion there. And because the isolation window is quite a bit wider than the high resolution survey scans, it'll still acquire an MS2 scan. And then because the ion trap can accumulate ions, you can still get a good enough signal to noise on the MS2 spectrum to identify something. But if you went and looked in the survey scan for this, it may not be anything that looks recognizable as a peptide. It might just be a single spike above the noise level that the instrument triggered on. And that happens with dynamic exclusion when you start working down at the noise level. So that's where these IDs come from. Whether or not they're going to be, particularly interesting in like a quantitative standpoint or not is another question. But you can recover some additional IDs from these things that are outside the windows. And you never find them with something like a 20 ppm search because they don't have an accurateness.
So I feel like what you just said kind of blew my mind. So you've got some fragmentation pattern of whatever, but what do you mean by like there's no accurate mass? So it triggered something, it let something in, it fragmented. You have the fragmentation that you can make an identification with. But what do you mean like there's no?
You don't have an isotopic pattern. Maybe instead of several isotopic peaks, you only saw one single spike above noise level. Mm-hmm. You don't know its charge state. You don't know anything about it. So the instrument does acquire those kinds of scans, and they're somewhat dependent on whether the instrument's got something better to do or not. So kind of the beginnings of your gradients or your end of your gradients where sample complexity might be lower. The instrument might have everything that it's already seen taken care of in dynamic exclusion, and then it's just kind of cruising the noise level to find other things to sequence. And the instruments, I think Therma has these methods where you can exclude 1+ ions, or you can exclude things that don't have an isotopic pattern. And then the methods often have the thing checked, and then like just take scans if you've got nothing else to do. So it's sort of this catch-all mode. Why have the instrument just sit there idle? It'll acquire scans of these very low quality precursors. And there's also another situation I think that occurs too, that you may have a co-fragmenting peptide, and the co-fragmenting peptide may score better than the peptide the instrument had a monoisotopic mass for. So you might have a Comet search where the second peptide scores better, and that's what the identification is. But because that peptide doesn't match the monoisotopic mass, you don't get an accurate mass for it. So I think there may be some potentials for sort of second peptide IDs to cause some sort of monoisotopic mass ambiguity.
Okay.
Yeah. Whatever they are, they seem to be there, and they seem to have very clearly nice score distributions, well separated from the noise scores. So whatever their origin is, I do think they're good quality IDs.
No, that took me a little bit on this afternoon to like process that one down. And you said that might be like an extra few percent like IDs. Yeah. Okay.
Yeah.
Cool.
And it's going to depend on the sample. It's going to depend on the acquisition method. It's going to depend on the instrument. But it's something here. It's kind of in there by default. So basically, everything that the instrument scanned and sequenced, we kind of look at the score distributions for. Some have accurate masses and some don't. Usually the ones that don't. Like I say, they're not super abundant, but they do look pretty solid. So once we've kind of worked all the way through the top tab and we've looked at all of our unmodified and modified ones and have set all of our score thresholds, then we can filter the data. And the console window now tells us that it's chugging through the data. And this takes a little bit of time here. Since we lost some time, we can do a Control-C out of this, because this data's already got. I've already got a filtered data here from the previous.
All right, so we're gonna put it in the oven and then
keep magic again.
Pull it out. So this generates some. So again on the histogram GUI, we just kind of went left to right on those tabs on top.
Right.
Okay.
So that'll chug through, and it'll make these filtered files. So what we have in the MSN files are all of our original SQT, MS2, TXT files, and they're pretty large. And then we get into the filtered one, we'll have the same set of of 3 files for every raw file, but now they're much smaller. That's associated just with the peptides that passed our score thresholds.
And can I ask one, because I should have said it. I mean, if you're running this pipeline and you just don't feel comfortable playing around with certain, I mean, so it's automatically going to set on those envelopes, it's going to kind of pick the shoulder of those peaks automatically. So like that's probably looks good. You can look at it, but honestly, it's probably good. And then those other thresholds are 1% FDR. They're probably fine too. So if you didn't know what you were, or you didn't feel comfortable messing with that, you could just click over and those defaults are acceptable. Like they're conservative. Yeah. Okay. Just clarify that.
But it's also a chance to actually look at the data and see what's going on. Like
Who
we saw
looks at
from the
data?
first Delta mask that there was a mask calibration issue associated with that. And then if we wanted to dig into it, we could load some subsets of the data and see that the May experiments have the one peak and the June experiment has the other peak. And we kind of understand a little bit that we've got a systematic mask calibration. And then we could either process the data in 2 chunks and combine them for the protein inference, or we could combine the score histograms and the mass histograms and just set wider windows. Both ways are kind of equivalent. You wouldn't get exactly the same answer both ways, but it'd be really close.
Cool.
Okay, so now the results program is going to read these filtered files and do the protein inference. So it asks us to select a folder with the filtered files. And it's read in all of the top hit summary files in that folder. We can list what's there. There's the 19 raw files. And now we need to assign them basically sort of a minimal experimental design. Here it's a label-free quant experiment, so each individual raw file is a separate biological sample, so we don't need to group together. We don't have like a multi-dimensional separation where we've got first dimension fractions that we need to combine in the biological samples. So we can also get a little bit of help information here. So just treat each file name as a separate sample. That's what we want to do. Just E for each. Now we've got all of our 19 samples specified as separate samples. We can list if we've got any leftovers. We don't have any remaining files, so we've assigned everything to something in the experiment. So we're done. Reads those files in and starts this reverse mapping. So these files come in as like, here's the peptide sequence and here's the protein accession that it goes to. So now we start turning it around. So every time we see a protein accession, we make a new data structure, and then we stick all of the different peptides that come to that protein. And then when we get done, then we'll have a whole bunch of proteins, and we can see all of the different peptides that are associated with those proteins, and then we can start doing protein inference.
And again, that will allow more than one peptide to go, or sorry, a peptide to go to more than one protein.
Right.
Okay.
So the first thing we're going to do is this, chugging through it here, we're looking for 2 proteins that have completely identical sets of peptides. And by identical, you always have to think about how the mass spectrometer works. So isoleucine and leucine are indistinguishable. So you have to make sure that you collapse those. But basically anything that's got the same collection of peptides associated with it, they're indistinguishable, and we lump them together. So that's the grouping step. And then after we've done that, then we start looking at conformal subsets. So if a protein with a small set of peptides is completely contained in another protein with a bigger set of peptides, then we can explain all of the peptides from the smaller set by the bigger set, and we don't need to report them. That's the parsimony principle part of the protein inference. So we remove all of those subsets, and each time it's sort of telling you how many proteins are on your protein list after each of these steps. Now we're writing the results tables. And we put out kind of a very detailed report that matches each raw file name. That's got all of the kind of common search score information and mass information in it. Then we make a peptide and protein summary file where we kind of collapse things more at the sequence levels that the biologist is going to be a little bit more interested in. And the pipeline uses 2 peptide per protein rule, which is, it doesn't do any sort of a protein ranking function and try to do any protein FDR analysis. It's just based on whatever peptides you pass through from your filtered file. It just maps them, does the parsimony analysis, and then requires 2 peptides per protein per sample. That usually provides pretty good error control and results in a pretty low overall protein FDR. So we had 9 decoy proteins out of 2,365 total proteins.
And in this case specifically, again, like for future whoever that's looking at this, you know, your GitHub write up on this data set and this analysis, like you go into that pretty well. Like there's in addition to this audio discussion, there's actually a pretty verbose discussion.
Right. So we got full analysis of this data set, and we've also got descriptions of all of the pipeline steps with the different yeast data set from the Gaiji lab. Yeah. So everything's pretty thoroughly described, and different data sets pretty much look the same when you run them through. So any of these examples are going to give you a pretty good idea how to do the analyses. So like I say, here we've got a detailed report for each of the 19 raw files. Then we've got our basic peptide and protein summary files that come from our first basic parsimony analysis. And then here, let me go over and look at this. We run an additional grouping step that's kind of interesting and a little bit different. So one of the things that happens if your protein database isn't kind of pre-processed, like there's these canonical proteins, FASTA files that you can get from Unipro, where basically each gene is represented by one protein sequence, but we know that there's alternative splicing and other sorts of things, and you can get multiple proteins associated with one gene. So you can get databases that are kind of one gene, one protein, or you can get databases that have multiple proteins per gene. And the bigger protein databases where you've got these multiple entries, we often don't in the shotgun experiments have see those peptides that distinguish the different isoforms for the protein. So even though the protein database is large and increases our search times, a lot of times we end up not having enough information there. A lot of those proteins are going to fall together in groups and not have distinguishing peptides. But sometimes they just have a teeny bit of distinguishing evidence. But it may be very minor compared to the total amount of evidence for those proteins. And we know that we don't have a 0% error rate in our PSMs, and these are big data sets. So those additional odd match to a protein could be an incorrect match. So maybe we don't want to rely on just a single peptide when we've got a bunch of shared peptides associated with 2 proteins as sufficient evidence to distinguish them. So what this extended parsimony grouping does is looks if we have just a little bit more than identical. So if 2 sets of peptides are almost identical, only have like 1 or 2 extra peptides in each, but they have a lot of shared peptides, they'll get lumped together. And if we've got something where it's almost a perfect subset of another protein, there's only like 1 or maybe 2 peptides that can distinguish it, but the amount of shared peptides is large enough to sort of outweigh it, they get removed as a subset. And then there's also a thing if we've got a little bit extra evidence on the things, but the amount of shared evidence is really overwhelming, it sort of groups those together as well. So we do those sort of 3 categories of grouping, and I call it extended parsimony analysis, just because these data sets are so large. And there's also the situation where you've got housekeeping genes and protein families where there's not much sequence difference between different members of the proteins, and you may have a real hard time distinguish them. And I described this too in the GitHub things, and there's a presentation there that also talks about this grouping algorithm. But basically for things like this protein we've got here, two proteins that had some shared peptides, we compare them, and we see that there were 35 shared peptides. The first accession had 25 unique, and the second one had 15 unique. That's enough unique evidence on that we don't want to group those proteins together. So they don't get grouped together, and nothing happens. And then we can also get situations like here, where we've got 47 shared peptides, only 1 unique for the first accession, 1 unique for the second. So that's a pseudo-redundant protein, 2 nearly identical sets. So those guys get lumped together. So this thing goes through and does that. And this stabilizes quantitative data, because usually the shared peptides are problematic in quantitative work. So if you don't lump those proteins together, then you've only got a handful of unique peptides that you can base the quant on, instead of using the entire set of peptides from those families, and you can get some really erratic results. But if you look at the accessions, the protein descriptions that are things that get lumped together, they make sense. They're almost always protein families or things that are isoforms of one another. But it gives a full report here. And you can also look at just the plain protein summary if you don't want the grouping. So it writes both out. I usually use the grouping stuff to main report that I look at. Hopefully that makes some sense. It's a little bit different than some of the other things. It's different than the razor peptide approach a little bit, and it's also somewhat different from kind of a clustering base that Mascot has, and it's also a little bit different than a protein grouping algorithm that Scaffold has. But they're all kind of based on similar concepts.
Yeah, I mean, I'm really glad that you're describing it, because I feel like that has been a change that's happened since I've been looking at stuff. This, how do you deal with protein groups, or what are groups? But I think that the thing, as you always describe it, is you're moving up to these levels where the benefit is at the quantitation.
Right.
And I always enjoy that, because that comes out in your discussions is as you move up the ladder of inference, it's almost like the data, I don't think you said smooths out, but that's kind of what you're getting at, is it kind of behaves better and allows for better downstream analysis.
Right. And we can open up there's descriptions of what kind of results files are written by the pipeline, and what the different columns are in the tables. But here we can see that the data is kind of your basic proteomics results table. So we got some arbitrary protein group number, we got our protein accessions, we got some kind of summary here of those parsimony analysis, protein inference, and protein grouping steps. So if proteins were identical, we see that there's other accessions associated with it. And if the proteins were similar, that means the things that we got together in the grouping algorithm, those get written at the bottom of the table. So we don't see any entries there.
Now, when you, sorry,
yeah.
where is the contaminants database just built in?
And that's part of the sort of FASTA file preparation steps with the FASTA utilities that I've got.
Cool.
So we typically bring down these databases and then add decoy sequences and common contaminants.
Got it.
And that's how they're sort of prepared for the target decoy stuff with common.
Another
back when this was all started, there weren't decoy databases, and then there wasn't consensus about how to make the decoy databases. And a lot of the search engines now have options to do the decoy stuff internally. And that's maybe safer than doing it externally. But this is still a little bit old school, so we kind of have to explicitly prepare the FASTA file for the common analysis, and we don't use the built-in decoy option in common.
Okay.
And then we have, sequence length, sequence coverage, protein description string. Then we get our spectral counts for the different proteins, kind of a basic thing. And if we were doing TMT analysis with the pipeline, then there's steps where we can add in reporter ion intensities associated with all the different samples. We've got total spectral counts for the 19 samples. Then we've got unique counts for the 19 samples. And then we've also got these corrected counts where shared peptides are fractionally split based on relative unique evidence. So this is a way to somehow try to come up with a little bit better total spectral count quantities for proteins if you're doing a spectral count comparison, which this pipeline is old enough that it was really developed for spectral counting. And then I've kind of used the same framework to just go in and add TMT reporter ion, extra hidden dimension for TMT support. But the TMT stuff is pretty much just added on at the end. So the basic function of the pipeline is pretty much the same, whether it's a TMT experiment or spectral counting like this. And that's pretty much it on the basic pipeline. Okay. Anything else you want to
Yeah. What?
cover? I mean, we've got all these extra things, the quantitative stuff. So we did a GitHub analysis just of this data, and we did a full spectral counting quantitative comparison on that. But there's also issues with these non-model organisms about trying to annotate things, orthologs, and have tools for those steps as well. But I guess it's a little bit far afield, but there's information here that describes all of that. And those would be the things that we would need to do to sort of finish up the analysis of this data, see what proteins are differentially expressed in disease groups. Then we need to know a little bit about the sample, what groups they're in. And then we also have to do something with these C-line annotations to get them into some form where we can compare the literature and stuff like that. Let me get back here and show where the analysis is for that. So we've got spectral counting experiment here. We've got C-line urine analysis of that with a description, pictures, and all kinds of stuff on that. And there's notebooks with the statistical analysis for the spectral counting comparison and stuff. So if you're curious about, kind of the end-to-end on this data analysis, that repository.
Yeah, I mean,
let's get back to
no, Phil, that was, and I should, again, like for future, I enjoy these like little phrases that you'll say, and it's actually like a whole other discussion. I know you've written up about like database preparation, why do you, de-redundant, those steps. The orthology mapping, that changed my life. So just as me publicly telling you again, that's a really big deal. And so I really appreciate that.
Yeah, we did lens stuff. So we looked at bovine lenses, mouse lenses, human lenses, and it's a question, how do you compare those proteomes?
Mm-hmm.
So that's kind of where that tool originated from. It's like I've got a list of mouse lens proteins and I got a list of human lens proteins, which line up, which don't. So the kind of brute force thing, the lens proteins are not very well annotated in kind of the usual things, because there's not very many of these lens specific proteins. They don't generally affect a lot of human health things, so they're not these well studied and characterized things. So they don't have any of the accession mapping information that you'd have for these genes. So you're pretty much stuck with having to go get up the protein sequences and then do the blast alignments to figure out what's what. But it's a pretty general purpose tool. Early on, there were a lot of protein databases available in non-model organisms that didn't have any descriptions. Everything was just an uncharacterized protein. And it's sort of like, well, how can I bootstrap some protein descriptions onto this file? So I'd use the same tool for that. You could just map to an annotated related species, and then bootstrap the annotation onto it, make a better FASTA file with some descriptions, and then your results tables were more meaningful for people to look at. So it's just had a lot of uses, and obviously these non-model organisms in comparisons are a pretty ideal case for them.
Yeah, you talk about like you can do so many things with it. Just last week we were using a plant database that, it was just like numbers. That was it. Mm-hmm. And what we did though is we blasted, or we took your tool and converted the results into what was published, which we thought what was on Uniprep, there were only like 300 IDs. Well, it turns out a lot of our results actually hit those. So although we don't know all of our results, we at least know quite a few of them. And that was just like a simple, click, click, boom, boom.
Right. You can do a Rapidopsis database, pretty well annotated.
Yeah.
So that's a common one for plant to try to figure out those other identifications.
Yeah. No, it's you came to, I guess, that talk. I gave some morning talk, and then you were there, and then you emailed me this. Mm-hmm. And I can't, it was like you dropped a bomb in my life. Like I had thought it was this horrible problem that I was going to spend 5 years solving. And then you're like, oh, I could do this today. yeah, it's changed all the mammal work and all the stuff that we do. So, um,
That
yeah,
was one of the better ideas I had.
yeah, yeah, no joke. And obviously, all the reporter ion stuff is such a big deal too. I was like jokingly going to ask you about stage races versus classics, but I don't. I think we went too far for that. Um,
I love the
uh,
women's races though this year. Almost all the classics had full women's races, and I bet the women's stage races are going to be a lot longer. It was like a 3-stage Tour de France for women in previous years, but
mhm.
the classics now they were doing 80, 90 mile races on slightly shortened courses. Dramatic improvements in the quality of the women's racing. It's really been fun to see.
It's one of the big proteomics hotspots is Ghent, and I
Yeah.
have talked to those guys in Belgium, and they
Mhm.
are huge cycling fans. So they, all the classics that happen around them.
Oh, yeah.
I think they actually all leave work, when
Mhm.
whatever is it, Flesh Leon or whatever, all these races
Right.
are there.
Yeah,
So
they're not allowing spectators this year, but normally those courses are just packed. I mean, it's
yeah,
hundreds of thousands of people on the courses. They love cyclocross too in Belgium. Cyclocross races are a big deal. That was really fun this year too. A lot of YouTube coverage, really good access to bike racing content. That's
well,
pretty dramatically different.
yeah, but no, it is my favorite thing to call you up because I get to talk mass spectrometry and last question, and then we'll wrap up after this. How are you so good at puns? Like, did you go to a special school? Is it just natural? I'm serious.
I've just
Like,
always tried to make crack jokes. That's, but over the years, a lot of humor can be very hurtful. So, sarcasm and mean humor, I'm quite good at that too. But over the years, I've decided I don't want to do that. So now I focus on puns because they're generally pretty benign and they usually don't have a very direct target. So,
no.
but no, I just read stuff and my brain just, substitutes what it sees or thinks or some phrase out of context just seems so strange.
Your pun game is definitely one of the best that I've ever been around. So I mean, obviously the bioinformatics and everything else is amazing. um, yeah, I mean, I think that's about it. So Phil, if we get to do this again, we might get to do a run through like with Reporter Ion Quant, which might just be like you said, like the back end.
Right.
But yeah, I mean, I appreciate you taking time out of your day. Yeah, and thank you again. I mean, for all the resources. So we'll put all the links to everything
Mhm.
with this, and yeah, just a big thank you.
Sure. Glad to try to give back to the community. So I've gotten closer to retirement age, and since I bike commute, I keep thinking I'm going to get hit by a car, and then everything that's in my head's going to go away. So I've worked really hard to try to get the bulk of what I've learned over these 18 years of proteomics up at GitHub to try to share it. So hopefully people find it and use it and tell other people about it.
No, I mean, thank you. I mean, yeah, it's a great resource for sure.
My pleasure.
All
Well,
right.
it's fun
Well,
hanging out. Good chatting with you.
definitely. Well, have a good day, Phil.
All right. Thanks. Bye.